Dear colleagues, my name is Anastasia Ilyina. I am a research assistant of the Laboratory of Molecular Mechanisms of Aging at the St. Petersburg Institute of Bioregulation and Gerontology. I am a PhD student of the Department of General Pathology and Pathological Physiology at the St. Petersburg Institute of Experimental Medicine. Today I'd like to focus on the molecular mechanisms in the application of short peptides under hypoxia conditions. Hypoxia is characterized by insufficient oxygen supply to tissues. Hypoxia is one of the stress factors leading to accelerated aging. Oxygen deficiency is accompanied by secondary tissue or bioenergy hypoxia, which is characterized by enhanced production of active oxygen species at the molecular level. Reactive oxygen species are involved in peroxidation of lipids in cell membranes that leads to cell abnormality. The formation of free radicals is a leading factor in the pathogenesis of ischemic injury in tissues and organs. It should be noted that administration of antioxidants into the organism play a crucial role in increasing the capacity of the antioxidant system and increasing the body's adaptive reserve under hypoxia conditions. Special place among these compounds belongs to geroprotector peptide drugs created and researched for several decades at the St. Petersburg Institute of Bioregulation and Gerontology. Today we are talking about two short peptides, myelin and epitone, which are active components of the dietary supplements terlutan and entelutan respectively. The EGR peptide, named as the pinolone, is an active component of the dietary supplement terlutan. The drug cortexine was developed based on a polypeptide complex obtained from the extract of the cerebral cortex of young calves. The dietary supplement terlutan was developed based on a polypeptide complex obtained from the extract of the whole cerebral cortex with white matter. The pinolone peptide was synthesized based on analysis of amino acid composition of polypeptide complex from the extract of the cerebral cortex of young calves. The IgG peptide, named as epitolone, is an active component of the dietary supplement endothelium. The drug epitolamine and the dietary supplement endothelium were developed based on a polypeptide complex obtained from the extract of the epiphysis of young calves. The epitolone peptide was synthesized based on analysis of amino acid composition of polypeptide complex from the extract of the epiphysis of young calves. First of all, let's talk about the effect of the EGR peptide on monoaminergic brain systems in hypoxia. It was evaluated the effect of the EGR peptide on monoamine content in rat brain in bilateral occlusion of carotid arteries as a model of hypoxic disorders. 18-month-old rats was used in this study, which are considered to be already old animals. The brain was initiated with a bandage of the left carotid artery and within a minute, temporary occlusion of the right carotid artery for 3 minutes with a subsequent 24-hour reoxygenation. The level of norepinephrine, epinephrine, dopamine, and dioxyphenylene were determined by a reverse phase variant of high-performance liquid chromatography. It was showed a significant decreased level of norepinephrine and an increased level of epinephrine both in the cortex and stem structures of rat's brain under hypoxia conditions. There is also a decrease in dopamine levels in the brainstem. Thus, there is a deficit of noradrenergic and dopaminergic systems in rats' brain under hypoxia conditions. It was detected an increase in epinephrine and dopamine levels in cortex and stem structures of rats that were injected with EGR peptide. The increase in epinephrine level in the brain of rats injected with EGR peptide may indirectly indicate an increase in its energy supply under peptide influence, because there is a decrease in energy metabolism in the brain with age. Accumulation of dopamine and dioxyphenylalanine was shown in the cortex of rats injected with the EGR peptide before the carotid artery occlusion. Increased norepinephrine content have been found in stem structures of rats under EGR peptide action. The change in the mediator ratios is only found in the cortex of rats, where there was a decrease in the norepinephrine to dopamine ratio. Thus, the EGR peptide has a modulative effect on the monoamine banks in the rat brain under oxidative stress. The following results were obtained from the study of the effect of the EGR peptide on caspase-3 activity and content of active enzyme form in rat brain under hypoxia conditions. Caspase-3 is the most abundant sustained protease in the brain and play a crucial role in synaptic plasticity due to its participation in the regulation of a number of brain signaling systems responsible for the establishment of interneuron connections, which is important for the implementation of adaptive responses to the aging process. There was an increase in the activity of caspase-3 in the stem structures and the content of active caspase-3 in the cerebral cortex and stem structures of 3 to 4 months old rats injected with the EGR peptide. Administration of the EGR peptide before acute hypobaric hypoxia contributed to the effect that the activity and content of active caspase-3 in the brain corresponded to the control level. It should be noted that there is a change in the activity of caspase-3 in the brain with age, which likely determines age-related differences in the body's response to the administration of the EGR peptide. Thus, preliminary administration of the EGR peptide promotes an increase in proteolytic activity and the content of active caspase-3, thereby preventing the development of the consequences of stress exposure in the hypoxia model. Next, let's consider the effect of melatonin and epithylamine on the content of protein and lipid peroxidation products in the cerebral cortex and hippocampus of rats' brain under acute hypoxia conditions. Important markers of tissue damage under conditions of acute stress are the accumulation of oxidatively modified protein and lipid peroxidation products, in particular malondialdehyde. Melatonin and epithylamine were considered as antioxidants in the study. The study was performed on juvenile outbred white male rats. The animals were divided into following groups: control, rats with the administration of melatonin or epithylamine, rats with modeling of acute hypoxia, rats with melatonin administered before hypoxia, and epithylamine before hypoxia. Acute hypobaric hypoxia was modeled in a modified low-pressure chamber by simulating the rise of rats to a height of 20,000 meters at a speed of 50 meters per second. The rats were kept on the altitude plateau until the moment of the second agonal inspiration, after which they descend to the previous zero altitude, restoring normal atmospheric pressure and the vital functions of the animals. The drugs were administered intraperitoneally 30 minutes before moderate hypoxia. Administration of epithalamine to non-toxic animals decreased the malondialdehyde content in the cerebral cortex and hippocampus. The administration of melatonin against the background of modern acute hypoxia reduced the intensity of free radical pulses in the cerebral cortex of rats to the control level. At the same time, only the intensity of lipid peroxidation normalized in the hippocampus. The content of protein peroxidation products in hippocampal cell not only did not decrease, but also continued to increase compared to the data in animals that were not administered melatonin during hypoxia. The use of epithalamine under hypoxic conditions significantly reduced the content of both products of oxidative modifications of proteins and the content of malondialdehyde in the hippocampus to the level of control animals. The results obtained indicate that the drugs epithalamine and melatonin reduce the intensity of free radical process in the brain under conditions of acute hypoxia, thereby significantly limiting the development of oxidative stress. However, epithalamine is more effective than exogenous melatonin in protecting hippocampal neurons under hypoxic conditions, reducing the intensity of peroxidation of both lipids and proteins. A more pronounced decrease in the oxidation of biomolecules in certain brain structures under conditions of oxygen deficiency after the use of epithalamine may be associated with the stimulating effect of peptides on the production of endogenous melatonin. Next, let's consider studying the effect of binolone on the survival of brain neurons during hypoxia. The study was conducted using cultures of cerebellar cells obtained from 20-day-old offspring of female vista rats. Oxidative stress was induced by incubating cells with hydrogen peroxide on the glutamate receptor ligand and methyl aspartate. Incubation of neurons with hydrogen peroxide or N-methyl aspartate caused a sharp increase in the death of neuronal cells. Moreover, in a suspension of neurons obtained from animals born from females under hypoxic conditions without the introduction of binolone, a twofold increase in cell death caused by hydrogen peroxide was observed. This value did not exceed 40% for neurons obtained from the offspring of females who were administered binolone. The initial level of dead neurons in the primary culture was also almost half as low in the case of neurons isolated from the cerebellum of animals obtained from binolone-treated rats. Thus, cells isolated from the cerebellum of animals born from rats treated with binolone are more resistant to oxidative stress. It was found that hypoxia leads to a sharp increase in the initial level of reactive oxygen species in cerebellum neurons. Against this background, the induction of oxidative stress in neurons causes an increase in free radicals, which differs little for hypoxic animals of both groups, that is, received and not received with alone. At the same time, the neurons of these animals differed significantly in their response to NMDA. Against the background of high steady-state level of reactive oxygen species, NMDA did not cause additional increase in free radicals in the neurons of the offspring of hypoxic rats, as it's observed in the case of intact neurons. In the neurons of the offspring of hypoxic rats injected with alone, a decrease in level of reactive oxygen species was observed upon incubation with NMDA. This fact shows that alone protects neurons. Since no analogy was found between the level of neuronal death and the increase in reactive oxygen species during the induction of oxidative stress, we can conclude that protection of cells from death is carried out by alone, not by directly limiting the production of reactive oxygen species, but by another mechanism, possibly associated with an increase in membrane stability, as we observed previously in the other experiments. In conclusion, it should be pointed out that the EGR peptide and IgG peptide are the active components of the complex dietary supplements serotin and endothelin respectively, are the effective antihypoxants. This is essential for improving conditions caused by the development of oxidative stress, including for the normalization of brain functioning. That's all I have to discuss in the present lecture, so thank you for your attention.